In our previous video, we learnt about one form of division by eukaryotes, mitosis. Prokaryotes, such as bacteria, divide and multiply by binary fission. Bi meaning two and fission meaning splitting. So what does this process involve? This is the process where cells make copies of its genetic material before splitting into two daughter cells. Firstly, genetic material of the parental cell replicates. Circular DNA is copied once, but the plasmids can replicate multiple times. The cell grows and DNA copy strands migrate to opposite poles of the cell. Step 2. The cytoplasm begins to invaginate for the new cell wall to form, so therefore the cytoplasm divides by a septum. Step 3. Once the cytoplasm has divided, two daughter cells are produced. Remember that each cell has its own copy of circular DNA, but can have a variable number of plasmids. Bacteria can multiply as often as once every 20 minutes under optimum conditions, such as right temperature, pH, oxygen, and availability of nutrients. So here's a graph showing the change in bacterial count over time. Population growth is monitored over a period of time and can be represented by a bacterial growth curve. The lag phase is where bacteria adapt themselves to their environment and synthesize RNA, enzymes, as well as other molecules. The exponential phase is where the bacteria grow very rapidly by binary fission. The stationary phase occurs when a nutrient is depleted, so the growth rate and the death rate of the bacteria population are equal. Then the death phase is when the bacteria die due to the lack of nutrients. You need to know how to estimate the number of bacteria in a population. To do this, you must use the mean division time. This is the average amount of time it takes for one bacterial cell to divide into two. Then calculate the division number, which is the number of times the bacteria have divided. And finally, calculate the number of cells produced in that time period. As each bacterial cell divides to form two, there is an increase by a power of 2 for each division, so the number of divisions can therefore be written as a power of 2. Let's go over a quick example to review this. A bacterial cell has a mean division time of 15 minutes. How many cells are produced after 1.5 hours? The first thing we can do is make sure we have the same units for calculations. So 1.5 hours is 90 minutes. We can then divide the replication period 90 minutes by the mean division time 50 minutes, which gives us 6, the number of divisions. To find the number of cells, find 2 to the power of 6, which is 64. You may have cultured microorganisms on an agar plate in your science practicals. But if you are asked to describe how one should prepare an uncontaminated bacterial culture, you must be able to describe aseptic techniques and explain why these steps are done. Bacteria can be grown in a culture medium which contains carbohydrates, proteins, minerals, and vitamins, which are required for growth. The culture medium can be either solid agar jelly on an agar plate or nutrient broth solution. Petri dishes and equipment should be sterilized in the machine called an autoclave, a high-pressure steamer. Work surfaces should be wiped down and sterilized, and one should wear goggles and gloves. Culture media must be sterilized before use. This can be done by heating. The inoculating loop used to transfer bacteria to the media must also be sterilized by passing it through a hot flame. All of these steps are done to prevent the growth of unwanted pathogens and avoid contamination of the bacterial culture. Once the agar jelly in the petri dish has cooled down, raise the lid of the petri dish slightly to prevent microbes from the air entering. The sterile inoculating loop is used to transfer bacteria to the media. A zigzag motion ensures that there is an even covering of bacteria. The lid of the petri dish should be secured with adhesive tape lightly. This ensures that pathogens from the air do not get in the petri dish. The petri dish should not be sealed completely, as oxygen is required for bacterial growth. Petri dishes should be stored upside down to prevent drops of condensation falling onto the agar surface. In school laboratories, cultures should be incubated at 25 degrees. This prevents the growth of pathogens that are more likely to grow above this temperature. However, in industrial conditions, cultures are incubated at higher temperatures so that they can grow faster. In school, you may have investigated the effect of antibiotics. These are drugs used to prevent growth and kill bacteria. Once the bacteria were cultured, paper discs are soaked in antibiotic and left in the petri dish. The antibiotic would then diffuse into the jelly. Antibiotic-resistant bacteria that are not affected by antibiotics will continue to grow in the agar around the paper discs, whereas non-resistant bacteria will die. The clear areas around the discs where the bacteria die are called the inhibition zone. The more effective the antibiotic, the larger the inhibition zone. The control paper disc is a paper disc that has been soaked in sterile water instead of antibiotic. The purpose of the control is to ensure that any difference between the bacterial growth around the control and the antibiotic paper disc is exclusively due to the antibiotic. The effect of different antibiotics can also be investigated by soaking paper discs in the respective antibiotics of the same concentration. The effectiveness of different antibiotics can then be compared by calculating the area of inhibition zones. Remember, the more effective the antibiotic, the larger the inhibition zone. To calculate the area of the inhibition zone, use the equation area equals pi r squared. Pi is a mathematical symbol approximately equal to 3.14. R is the radius of the inhibition zone, so you can measure the diameter of the zone and halve it. Square it and then times by pi. Let's apply this equation to a practice question. Here we have 5 different antibiotics soaked by a paper disc used in the lab to investigate the effect of bacterial growth. Here are their respective diameters. Now, from these results, which antibiotic is the most effective? Show your working and explain your answer. Pause the video and have a go now. First thing you should do is calculate the area of inhibition. Remember to halve the diameter to get the radius for calculating area. From your calculations, you should see that D is the most effective antibiotic because it has the biggest area of inhibition. This means that more bacteria are killed and therefore are less resistant to the antibiotic. We hope this session on binary fission has been useful. See you next time.